So now we are speaking with Dr. Luis Zia, who's an assistant research professor for CU Boulder Aerospace, and he specializes in the intriguing field of bioastronautics, which is studying how biology and space interact. His interests span everything from gravitational microbiology to biomining and bio-based resource utilization. His work asks questions like how can we protect our astronauts from bacterial contamination? Or how can microorganisms help us mine metals from asteroids? Dr. Z is also part of the Artemis program, leading an investigation to use yeast to study the radiation environment of the moon. This all sounds so fascinating. So thank you so much for joining us today.
Thank you for having me. I'm excited to be here.
Great. So I figured we'd kick it off with probably kind of an easy question, maybe.
I'm an Aquarius.
Perfect, me
That
too.
it?
So bioastronautics is not a term that we really hear very often. Can you kind of describe what exactly that means?
I hear you. Yeah, that's kind of something that you hear rarely, definitely not at the grocery store. The very first time I heard the very short and to the point definition was from Dave Klaus, a professor here at the University of Colorado, who spearheaded the bioastronautic focus in the aerospace engineering sciences department. And now it's grown a lot. When I started my PhD over a decade ago, we were a handful of graduate students in bioastro, and now it's a very large group, which is very cool. And the way he summarizes it is bioastronautics is the study and support of life in space. So really, there's different aspects to that very short sentence. When you think of the study of life in space, it is not the same thing as astrobiology, where astrobiology scientists are looking into, among other things, potential origins of life. Is Earth the only place where life exists? How may life look like in other places? Whereas the study of life in space, part of bioastronautics, is more of bringing Earth life to space and studying it, see how it changes. And when you think about it, if you do a very quick time travel through Earth history, about 4.5 billion years of history, we know at least the oldest fossils for which there is agreement that they are, they were of live organisms, are about 3.5 billion years ago of microbial fossils, in fact. And so you think about it, what has changed on our planet in 3,500 million years? And the composition of the gas has changed, the temperature, oceans, the salinity, the pH in the ocean, all these things have been changing, and life has evolved adapting to those changes, but something has remained constant, and that's gravity. Now, you can send Earth life to space and investigate what happens to cells and organisms. In other words, you can treat gravity as an independent variable. You can study what happens up there. And in fact, just growing stuff in space lets you see things that don't happen on Earth. And I have a bunch of cool stories. Of course, I'm a big nerd in this, so I think they're cool. Maybe nobody else does, but they're pretty exciting, I think. Not only because they're like wow moments, but because they can help us find solutions to problems on Earth. And that's the reason for which we do cancer research on the International Space Station. That's why I focus, for example, on bacterial research in space, not only to enable safe human exploration of space, but also because we're seeing things that we have never seen on Earth that could potentially help us, for example, to fight drug-resistant bacteria, which is a problem that in the 1990s killed about 10,000 people a year. Now it's over 100,000 people a year that die from drug-resistant bacterial infections. That infections that we could have cured with drugs before, but these bugs have just acquired resistances that we no longer how to go around. And the other part is the support of life in space, of the definition of bioastronautics. And when you think about it, think of any sci-fi movie you like. I'm gonna go with Spaceballs because I think it's awesome and because I just watched it. And just think of what's in a spacecraft that enables humans to be in it, right? ECLIS, Environmental Control Life Support System. What does it do? It provides you with fresh air. It removes carbon dioxide. It provides you with clean water. And of course, you're not going to bring all these big tanks of water to long missions. So you want to have a closed system as possible. And what closed system means is that you are recycling the whatever you produce, and this is done on space station, the urine, the vapor from the sweat, all of this is reprocessed, and then it's filtered, and then it's potable water, and it's also water for other systems. So very long answer to tell you that the definition of bioastronautics is the study and support of life in space.
So did you have to do this, kind of a background question, but how much medical knowledge have you kind of been forced into in this field? Is it more just science, or do you actually get into a lot of the medical field?
Good question, and I'll split it in two parts, and I'm doing so more to encourage people to get into this field, even if they don't have any bio background. For example, my undergrad from Guatemala is in mechanical engineering from UVG, Universidad del Valle de Guatemala. My master's in aerospace with a focus on thermofluid systems. Then I worked as a heat transfer engineer. And then I did the leap of faith through bioastronautics program into sciences. And now what I do is microbiology. And so the way I would answer that is that you do not are needed to have a strong background in bio because you, or medical, because you will acquire it. However, if you do have it, then great for you. And that's one of the messages I would have for the next generation of folks who may be listening to this is the multidisciplinarity of life. Real life is not like, especially when you're in high school, like you're about to graduate and you're like, oh my God, what am I going to study? And you think that if you go for this one thing, that's the one thing you're going to be doing for the rest of your life. Like Fred Flintstone, like he had one task to do and he did it every day, every week, every month, every year of his life. That's not real life. And if you have the ability, if you have multidisciplinary background where, for example, you learn a little bit of biology and medicine and engineering or computer, you name it, then you have a stronger footing to learn all these kind of things. So during the bioastro program, then you learn stuff. Most of the bioastronautic students at the University of Colorado have an engineering background, but it's not exclusively for engineers. In fact, there's a lot of people who come from a biotechnology or molecular biology background. They have to take a few courses to catch up, and then voila, you go with bioastro.
That seems to be kind of a theme actually in a lot of these interviews that we've done, and going way back to the very first one with the director of the planetarium, John Keller, who didn't initially set off to do astronomy as a scientist, and after a couple years of figuring out that that's what he wanted to do after his undergrad and some teaching, he came back and got a master's, which was basically just a couple of supplemental courses that really set him up to be able to do that. So yeah, that's a great message that you're not locked into what your undergrad was, or whatever you may choose to do right out of high school. Jumping right into your projects, Tara and I were talking about how your website, looking through your website is like it looks like reading a sci-fi novel or something. Like all of these different projects that you're working on are just crazy. So one of these projects in particular is examining how biofilms behave in space. And biofilms are groups of bacteria or fungus that stick together, like the kind that you find in your shower or on your teeth. You can correct us if that's wrong, but that's our understanding for everyone listening. That's a biofilm. And much like we never think about them in the bathroom until it's too late, I've never considered that they would grow in space too. How are you conducting this experiment? And are you just characterizing them at this point, or are you also actively testing ways to mitigate them?
Yeah, no, you got it right. Except for you said biofilms grow in your bathroom and your teeth. The correction is they don't grow in my teeth. All right, I'm exceptional. Yeah, that's actually right, and that's why we brush our teeth, all of us. Because even if you were to be just rinsing it with like mouthwash, that is not enough of a mechanical stress to remove them from our teeth. They are biofilms. As you mentioned, bacteria and fungi, mostly bacteria. Otherwise, fungi, when they're forming biofilms, are usually called molds. Technically, you can use both. But these bacteria, for example, in people's mouths, in our teeth, they secrete these substances that allow them to adhere to a surface. And once they're there, good luck. You need really hard brushing. And that's not only a problem for astronauts' teeth, but it's also a problem for, well, we were mentioning before of ECLAS, environmental control on life support systems. what keeps the astronauts alive in space. And let's go for another quick time travel. And now it's about space stations on orbit and what biofilms problems they have had. So the first big space station, the Soviet Salyut 6, it had several biofilm problems. They saw it forming inside pipings and behind panels. The Soviet Salyut 7, they had it on the water recycling system. That's a place you don't want it. Rubber of hatch locks. So if you think about it, the doors, if you will, in space stations or spacecraft, they have it's like an O-ring, right? It's a seal to make sure that gases are not going anywhere. Now, biofilms are a problem for two reasons. One is because of the health problems they can bring. In fact, most of bacterial infections in humans have a biofilm association to them, meaning that bacteria tend to be in a biofilm state when they are causing the disease. And the other one is because they can degrade the material. So they can basically eat away the plastic or metals upon which they are growing. They can be acidic, they can be corrosive. So if you have this kind of stuff growing on an O-ring that keeps your air in your spacecraft, that's not a good thing, because now you have little pads of air leaking. And if you have it in your water recycling system, as I mentioned, there's also this medical aspect of it can get people sick if it's the wrong kind of bug. So also you don't want it anywhere close your water, right? Uh, um, and then if we keep on the time travel, the Soviet/Russian Mir space station, it had it in a bunch of different places: air conditioning, the oxygen electrolysis block, the water recycling unit, even on an extravehicular activity suits headphone. This is the cool suit that you saw. What was that movie, Gravity, with all that wrong stuff, but also some pretty cool looking stuff. Yeah, so those are the EVA suits. On the Mir space station, they had biofilm growing in one of the suit's headphones. Um, and very interestingly, one case was on a navigation window. So they had this quartz window that the cosmonauts would use to look out and see the spacecraft that was arriving, the progress with cargo and stuff they needed. And they would kind of like play video games, but real life, and bring in it. Now, that navigation window was going through problems of what eventually they later learned was a fungi called Penicillium rubens. It was called Penicillium chrysogenum, but it got its name changed. It wasn't cool enough, so it went for the rubens. And you don't want your navigation window not to be perfectly clear, right? And you don't want that jeopardized by biofilms. And so it's been a problem for a very long time. In fact, there's this really cool story of when astronauts, US astronauts, were able to go to the Mir space station and telling stories. And also, these stories also coming from cosmonauts of, for example, a really bad stench. And people are like, what the hell is that smell? And you are living in this very tiny space. You don't want to be smelling that kind of stuff. Like right now, we're all quarantined, right? We take our trash out, because imagine being quarantined and smelling that stuff all the time. And so they find out it was coming from a panel. So they remove the control panel and they see all this biofilm growing on the back. Now, that's bad for a lot of reasons. But remember how I was saying that biofilms can degrade polymers, the plastic, for example, that covers cables and wires? Big no-no. You don't want another fire there, right? So that's another kind of problem of biodegradation, of how these bugs can cause problems. And of course, this is true not only in Russian and Soviet space stations, it was also true, and it's also true still on the International Space Station. And that's the reason for which NASA is currently supporting research in this field, because especially there's a challenge making sure there's no biofilm growth on the wastewater tank, which is part of the water processing assembly of ISS Cyclus.
So are these biofilms, do we know where they come from? Or are they just a byproduct of people being people and full of bacteria to begin with? Or is it something that we can contaminate out before we launch?
Yeah, these are good questions. And those are the questions that NASA headquarters has been asking themselves, right? And when you think about it, there are more bacterial cells on you, listener, than your own cells. So actually, there was an old paper that said it was more than tenfold. That paper a couple of years ago it was reviewed, and they found out that was not the real ratio, but still it is true that there are more bacterial cells in and on us than our own cells. So wherever we go, they go. Wherever if we go to Mars, we go to, future Europa stations, bacteria are going to be traveling with. And as I mentioned, most of them form biofilms. A lot of them form biofilms. So that's where they come from. We bring them. Even if we were to sterilize surfaces, which in a lot of cases, they are very, very clean when things are sent up to station. Humans are going to be bringing stuff with them. A separate thing, and I know you've talked about this in previous podcasts, is making sure that we don't bring Earth stuff to other bodies, celestial bodies, right? That's a different story. But here in this case, we don't want it because of these problems that I was mentioning. And yeah, that's where they come from.
And I imagine problems that we're having with them in the space station will definitely translate if we're, living in little habitats on the moon or on Mars or something. It's a very comparable situation there as far as being stuck in a small space.
Yeah, and that's the other thing. We don't know. We're seeing, because so the space biofilms experiment has launched to space station last year on Northrop Grumman's 12th commercial resupply mission, and it already returned on SpaceX 19. And we're looking at the biofilms right now. A PhD student, Pamela Flores, she is at the microscopy core at this moment exactly, taking images of the biofilms. We flew 144 samples to space, 144 samples remain on Earth, and we're studying differences in their growth. And we have basically no knowledge of how biofilms will be different on the moon at 1/3 G or at Mars 1/3 G, or on the moon 1/6 G, where G is Earth's gravitational pull. So we got lots to learn, that's for sure.
Is there something about biofilms and their structure that make them hard to get rid of? Because you mentioned that, say mouthwashing, right? We find biofilms on our teeth, and simply mouthwashing is not enough to get rid of them. Is there something about like their structure that makes it so that some antiseptic fluid or or whatever Clorox wipes isn't enough to just kill all of that bacteria and mitigate that problem?
Yeah, there's a lot of different variables to the equation of that question. There is of what is it that they excrete that makes them these adhesins that make them be capable of adhering to stuff. What is the topography of the surface they're growing on? And there is a really cool study of a biofilm investigation done on Space Shuttle in the past, where they had the biofilms growing in space and they had Earth controls. And in Earth, if you're a bacterium, these biofilms look kind of like a carpet, like a mat, right? They are just flat. But what they saw in some of the samples grown on space is that if you were of the size of a bacterium, it would be like you walking on a forest. They call them the column and canopy structure that had not been observed on Earth before, where basically there are these trunks of biofilm. And on top of the trunks, there's this canopy, there's the mat. And then the question is, what impact will that have on all the different things we were talking about? Like your question, is it going to be harder or easier to remove? From the medical perspective, does that change their virulence or their capability to cause disease on an astronaut? And from the materials perspective, is their corrosiveness and biodegradation capabilities change because of this structural change of now you are walking on the forest of the biofilm?
That's kind of a terrifying image that you have this like this like three-dimensional structure of of bacteria, this
Yes,
forest.
probably smells worse than that panel.
Yeah, right.
Well, kind of moving forward, I also want to ask you about your mission on the Artemis program. You're a co-PI for this. We talked about Artemis a little bit last week with Dr. Hain, and you're particularly studying how radiation in the moon's environment could affect cells and DNA using yeast. Can you kind of tell us a little more about how this works? Because this is really strange.
Yeah, right. Yeast. That's what we use for making bread and beer. Yeah, actually, when you see the genome, which is the genetic code of yeast, over 70% of their most important genes are also found on the human genome. So that makes them a really good model organism, is what we call it. For example, imagine if I want to have really strong statistical data that I can do studies with. I would need to send thousands or millions of humans to deep space and analyze changes, right? However, in a tiny little vial, I can send millions upon millions of yeast cells, and each of them have their own DNA. Each of them have their own genome, and that allows us to do very massive studies with genes that are also found either directly on the human genome or they have equivalent genomes. And that's the kind of like the reason. And why on Artemis? So Earth is protected by the magnetosphere, right? So if we were to cut the Earth in half, which I hope nobody has the powers to do because that would be a bad day, you would see something like an apple, where the Earth is a circle in the center, and then you would see these two kind of ovals that go outside of the circle, and that's our magnetosphere, right? It has multiple layers, and this magnetic field protects us from some of the solar particles that are always irradiating our way, but also from galactic cosmic rays, which sounds very sci-fi, but it's really real. And that's the reason for which we're not getting cooked, right? But once we are beyond the Van Allen belts, astronauts are going to be exposed to that high radiation environment. And we haven't had an opportunity to send a controlled biological experiment beyond the Van Allen belt since 1972 on Apollo 17. So when NASA put out a call for proposals, I was shaking of excitement, and of course, I partnered with Corey Nislow. He's a professor at the University of British Columbia, and he has this really cool things called deletion series and overexpression series of yeast. And if think of the genome of the genetic code of anything, say in this case yeast, of like a bunch of Lego pieces, making a little train. In this case, it's about a 4,000 Lego piece long train where each Lego is one of the genes. With what he does with his deletion series is he makes, imagine, 4,000 copies, and on the first copy, he removes one of the Lego pieces. And instead of that, he puts a little flag. And the next one, he removes the second Lego piece and puts a flag. Basically, it's called a deletion series because each of those mutants has had one gene deleted. And the overexpression series is the opposite. Instead of deleting one gene, he puts a bunch of that gene in that space. And what we're going to do is at BioServe Space Technologies, which is where I work, it's a nonprofit research center within the University of Colorado. We build hardware and we help we help scientists send their experiments to space. We train the astronauts, we develop the hardware, all the NASA safety support documentation, etc. So what we're doing at BioServe is building the autonomous hardware, which of course, this is space, so it has to have an acronym. It's called PLASM. And PLASM gets no data, no power, no communication, no nothing from Orion. And still, we need to be able to know when we're past the Van Allen belt and activate the experiment. That's what we're going to be doing, activating this deletion and overexpression series, then letting them grow under the microgravity and high radiation environment of deep space. And then we're going to make a correlation of which of the mutants had higher rates of survivability than the others. And this allows us to make a study of the effects of both microgravity and high radiation on DNA damage. Another really cool thing is that yeast has DNA repair mechanisms that are also similar to what happens on humans. We don't get cancer as often as it could be, thanks in part to the DNA repair mechanisms that we have. Imagine this DNA strand, and there are these little molecular robots that swim through it, checking for any inconsistencies. And when they found something that, like, oh, wait a minute, this should not be a G, this should be a C. And they recruit these other molecular robots and they fix it. Well, and you want to make sure that this repair happens before the cell splits in two. If the damage was not repaired before the cell splits in two, now that's going to be part of its genome, and that those two cells are going to become 4, 8, 16, 32, 64. That's one of the bases of cancer, when this mutation just takes over. So it's pretty important to understand if we are going to have higher rates of DNA damage in deep space. We know we will, but we don't really have that problem quantified. And the other aspect is we also want to see if the DNA repair mechanisms are acting in any different way.
So what I'm realizing as we're talking to you is that this is going to be a very difficult episode to cut because I will want to keep every word that you're saying because this is so interesting.
Don't cut
So
it, Colin. Don't cut it.
I don't do it. So um you very briefly mentioned at the beginning of right when you said, oh yeah, yeast, like we bake bread with. Is that the same yeast that you're sending in these experiments? Is there a difference in the different kind of yeast cells that
Yeah.
you're working with?
Yeah, there's a whole bunch of different strains, we call it, of yeast. For example, you can have, say, E. coli is a bacterium, that's the species. And then there's a bunch of different strains, which are smaller differences in the genome, but they're basically the same cell, the same bug. In this case, yeast, there's a whole bunch of different strains. If you go to a brewery, nobody would want to do that because that's super boring, right? But if you go to a brewery, each of them has different yeast strains that they use for their beer. That's part of their secret recipe because they will behave slightly different when exposed to the same constituents of an ingredient. What we're using is basically a wild type, which means it's something that is wild. It's something you would find in nature. But then Corey and his team go ahead and make that deletion and overexpression series.
So are there any future plans to try and maybe like brew beer on the moon? Is that a thing we're thinking about?
Oh, you know how bacteria go wherever humans go? Beer and bread are gonna go wherever humans go too. That's a fact. Oh, I'm sure. Actually, at BioServe that we've flown you so many times, and in this we have this hardware called the Fluid Processing Apparatus (FPA). It's a little glass tube, and they go inside this other thing that's kind of like a glass, like a cup size. Of course, today I'm drinking my water. I just found my daughter's princess's cup, but it's about this size, and we've flown and operated FPAs over 6,000 times in space in over 25 years about it. And some of those experiments were with yeast, and some of them they simply did the same process they do. So in principle, that has already occurred in space, but not in a commercial way. And I'm sure that's just a matter of time before that happens. Yeah, I'm looking forward to trying it actually.
I wonder if there will become a new kind of branch of brewing in microgravity. It's a new meaning for the name microbrew.
Oh
It's
yeah,
brewed in microgravity.
microgravity brew. I like it.
I like it. Now, you also mentioned having to train astronauts to do these experiments. This is not necessarily something that they're already planning on doing necessarily. A lot of them, come from aerospace backgrounds and pilots and military and that sort of thing. So how is that going? Do you have a lot of trouble training these pilots to be scientists or that sort of thing?
And I'll say that bioservices is a small group of people, and we have a crazy amount of experiments going to space at any time. For example, in the next two rockets going to a space station within a period of time of a month, we have 10 payloads that we are supporting. And despite of that, we're a small group of folks, and there's this one person. She is our operations lead. Her name is Shankini Doraisingam, and she's the one who goes to Houston to train the crew. And yeah, that question comes up a lot. And what I've heard her say is usually that they're astronauts for a reason. They are rock stars. Some of them indeed have a background that may not be in science, but they're fast learners. They pick up. And of course, you have kind of like your favorites, which you shouldn't. But for example, I remember working with in payloads with Peggy Whitson and Kate Rubens. Oh my God, they're robots. They are so good. They get the job done so well, so quick. It is really cool to see them work. One of them was with Peggy was a cancer payload, and we flew human adenocarcinoma from a 26-year-old Caucasian male. And this was for two startups that were funded through CASES, the ISS National Lab, and they're called Oncolinks and 3D Biosciences. And they were at them, among other groups, they want to grow cancer tumors in space. And why? What's the benefit of doing that, right? And when you start growing these cancer tumors on Earth, as soon as you have enough cells, they're going to start piling on top of another. And when they do that, the one that's on top just falls down it just becomes a flat mono layer, one single layer, like a pancake. Whereas in gravity, that doesn't happen, and these three-dimensional structures are built up, and that best resembles what happens in the human body. So now if you have a molecule that you want to go through the FDA process with to see if it can become a drug, then you have a target that is going to give you less false positives or false negatives, because it actually looks more like what you're trying to target on the human body. Total deviation. You're asking me about astronauts, and I talk about cancer research in space. Sorry, but that kind of like the point there is, and that's something that us space junkies, I think, when we go to like Thanksgiving dinner and they tell us, so why are we spending all this money on space? Some people think that you're actually putting the money in a suitcase and sending it on a rocket. In reality, and some people think even if they work in this field, they can't come up, and this was me too, right? That you can't come up with really good examples like, oh, Velcro, yeah, not really, of the benefits. But in reality, think about these kind of cases. For example, we're testing potential drugs against cancer. Another example is osteoporosis, right? Like right now, like if the listener of this is sitting, or even if they're laying on the bed, there's some mechanical stress on their bones that keep them healthy. This doesn't happen with postmenopausal osteoporosis, where you can have about 1.2% bone mass loss a year, meaning that every year you've lost about 1.2% of your bone mass. In space, if you don't work out, it's about the same rate, but a month. So it's an order of magnitude worse. So yeah, sure, that's a problem for space agencies to make sure that astronauts are going to be safe for long-term human spaceflight, but it's also an opportunity for us to test potential molecules to see if they can become drugs. In fact, that's an example of this is the drug called denosumab from Amgen. And this was a couple of experiments were flown by NASA Ames, supported by Bioserva, of course, Amgen, it was their drug, and a bunch of other institutions. And it was approved by the FDA in 2010. In 2012, it sold over $1.2 billion. And it's a drug that can help people stop the process of bone reabsorption. As we grow older, the rate of growth of bone absorption is higher than the bone generation rate. And that's why for little kids, it's the opposite. They're just generating bone like crazy. So they break a bone and in no time they're fine, because the rate of bone generation is higher than the bone absorption rate. That's not the case when we're older, and these kind of drugs help people on Earth with those cases.
I think, you stopped for a moment to say, sorry, this is a space podcast, and here I am talking about, all of this stuff that's not that. That's the beauty of these conversations, and that's what makes talking to people like you so fun, is that you have this perspective from outside of the only space circles that adds a lot of really cool stuff. So there's no need to apologize, and please don't worry. I mean, go on those tangents. That's why we're here. That's what's so fun. So moving forward in some of your, another one of your sci-fi sounding projects that is sci non-fi, you are involved in something called biomining, which to our understanding is using organisms to mine for metals like iron on the moon. What sort of organisms are these? How does it work? What are the 44 endangered elements that you mention on your website? Also, quick plug to your website. Anyone listening, the website is awesome, so check it out. You can find it if you just search like colorado.edu space Luis Zia, and we might hyperlink it somewhere. It's a really cool website. Anyways, back to you, Luis.
All right, thanks. Yeah, maybe every time we say the word space biomining, John should put like a cool sci-fi background on the podcast. Yeah, it sounds really sci-fi. In fact, it is not. And something that a lot of people don't know is that between 5 and 15% of gold and copper that is mined on Earth comes from biomining processes. So I'm sure that the first person who thought of biomining on Earth probably got laughed at a lot, because that was totally sci-fi, more on the fi. And but in fact now it is not, because we use it a lot on Earth. So where the novelty of this is implementing these kind of processes for in situ resource utilization. So making sure we can live off the land as we go to cool places like the moon, Mars, and beyond. And there's this one bacteria called Shigella oneidensis, and it has the capability to transfer electrons with minerals that are outside of it. So it's called an exoelectrogen. And what it does, it can help us reduce iron. So iron present on the regolith, on the dust, it can be oxidized. And imagine it's called iron 3. And then Shigella goes there, it eats, it does its thing, and it transformed that iron 3 that was in the mineral into iron 2 that is now in solution in a liquid that you can then segregate from the rest. And that's how you could potentially extract minerals. We're starting with iron in the lab because that's something that there's a lot of research on how Shigella does that. But what we're doing here is that we're growing on a spaceflight hardware, those FPAs and gaps that I was talking about, and we are exposing it to regolith simulant. So we don't have, you can't go to a grocery store and like, hey, can I have a pound of lunar dust? But there's this really cool lab at the University of Central Florida, where it's actually where I did my master's, go Knights, and this called the Class Exolith Lab. And they actually synthesize regolith simulant from different places of different bodies. And that's how we have asteroid, lunar, and Martian regolith simulants. So they have what we understand are equivalent constituents. That's the same ingredients, right? And we're growing the Shigella with these different kind of regolith simulants in flight hardware, like we would do in a potential spaceflight experiment, which we're still not yet planning. We're just learning the ropes here. And the idea is that eventually you can have these tanks where the bug is happy, you're giving it the food it wants, it can do this electron transfer, and then it will give you things that you can need for other things. For example, if you have this bioreactor where you just put a bunch of dust and these little things with bacteria and its growth medium, and on the other end, you're getting purified iron, then you can feed that to 3D printers and you can build structures, you can build parts. And there's a bunch of metals that you can do. And you mentioned the 44 endangered elements. And that's the idea that this could support not only space, having folks out there in space, but it can also be brought back to Earth. Imagine how cool a world would be in the far future where Earth is declared a no mining zone, like no heavy industries. And Earth is a living zone. We could do mining off asteroids, and that's where we get all the stuff we need, or at least part of it. And biomining could be a part of it as well, because it can help you potentially extract some of those endangered elements, which are the elements for which we know there's going to be a supply. There's not going to be enough production of them in the upcoming years.
So is it like this one strain of bacteria is good for all of these elements, or are there different strains that are good for different elements?
That is the question. We need to learn which one do you want to use for which application. And it's definitely not a one solution for all, but it's more of the process. Can we implement this process with what we're talking about? How stuff from Earth grows differently once you have microgravity, for example.
What would be, and this might be like a high level question for just a standard, podcast episode, but we'll go here. What is the process by which you use this bacteria to transform the element? So, you mentioned, you have this iron 3, you introduce this species of bacteria to it, and then it does its thing, and then out comes iron 2. Like what does that process look like? Like if I were the astronaut that you were training to go do this biomining, what would you tell me to go do up on the moon? Do I take a vial of regolith and then put this?
I'll tell you, Colin, take me with. I can tell jokes and cook and maybe make beers.
Perfect.
With.
You're on the team.
Yeah, so actually, it like everything, it begins like a very low TRL, technology readiness level. So it will begin as experiments. We need to learn what we don't know. Once you have found the answer to a question, that generates 2 new questions, and that's how things work, right? So likely would start with these biosource FPAs that I was mentioning, and the long-term vision is that these are going to be large bioreactors. Imagine the fuel tank of a rocket, where you have this automated process of a robot just sieving the regolith, which is filtering to make sure it's the right size, and then drops it there, then it closes it, and in goes the bacterial culture. And the bioreactor needs to provide it with the right temperature and pressure. And and then you just let it sit. Actually, that's kind of when you see how biomining works on Earth, it's just a pile of dirt, a very large pile of dirt, sometimes like stories high. And on top, they just have this little tube that is dripping this icky looking water. And at the bottom, there's this little kind of like a little channel that is collecting the icky air water. Now it looks way different. And it's because all that the bacteria they have in that pile of dirt has been processing it, and now what is spitting out on the bottom, it has whatever mineral you're looking for, example copper, in a different state that makes it easier to separate it. So I in a long-term future, I wouldn't think there would be much crew, um, crew need, but they would need to maintain the hardware and stuff. And at the beginning, it will be experiments, so for sure you will need astronauts helping you.
Very cool. Thank you. I look forward to my time as an astronaut on the moon doing some biomining. I think that's going to be a lot of fun.
That sounds really awesome. So we did want to kind of here towards the end ask you a couple of little more personal questions, just kind of about what you do in your journey and how you got here. And on your CV, it talks about you started your education in mechanical engineering and then aerospace engineering and then bioastronautics. So have you always had this interest in space or space science, or is this something you kind of gradually worked your way into?
Oh no, it wasn't my genome. For sure, it was there from the beginning. And I'm from Guatemala, as I mentioned, so we didn't have the the resources for me to come to the States, so I wasn't sure I was going to be able to. I started as a not a great student, meaning I was a terrible student, and then when I realized that I wanted to do space stuff, and in order to be able to come to the States and do this, that I needed a scholarship, that's when my life changed. I did skateboarding for a living, so my priorities was first was skateboarding, and then a number 2 was skateboarding, and number 3 and 4 skateboarding, and 5 maybe if I had time left, it was school. So when I realized how things worked, I changed my life and really focused on studying and trying to be a better student. This is in high school still, middle school, high school. And then I wasn't able to get any scholarships because I had all these years of slacking, right? I ended up well, but I had all this history of not doing great. So I learned German as a plan B, so that I could apply to Germany. I learned Portuguese as a plan C in case I couldn't come to the States or Germany. I would try to go to Brazil to study aerospace. I did an undergrad in Guatemala to get my stuff together and get that degree, and that's how I was able to come to the States as a graduate research assistant, where I worked helping with research, and that was what pays for your tuition, basically. And that was in petroleum engineering. And that's because after my undergrad, I still couldn't find a way to come outside of Guatemala to study aerospace. So I got a job as a mechanical engineer in the oil industry, and actually had nothing to do with oil. All I was doing was Excel, and that's what helped me get my foot on the door. Then I did aerospace. Then I couldn't really get anything into aerospace because I wasn't a resident, a citizen. So I got a job as a heat transfer engineer. And then I went back to school for a PhD here at the University of Colorado Boulder. Now I'm a citizen and I'm able to work on all this cool stuff. So life takes you different ways. Life really is a zigzag. And that's kind of the message I would say to younger folks. Just keep an mind open, plan for a plan B. The more tools you put in your toolbox, languages, software, stuff, the better for you.
That is an awesome story. Like, just that whole progression that's just fascinating.
I love hearing these too, because it seems like pretty much everyone that we've talked to, nobody has had a real like linear path of how they got from A to B to C. Everybody's kind of been, started here, and we had some musicians and we had some bio folks, and everybody kind of winds their way. Nobody really just goes straight. And I think that's awesome.
So you mentioned that you first of all, you said, well, I learned German and then Portuguese just to have those options. Was that like a casual process for you? Like, just decide, hey, this week it's going to be German, next week I'll do Portuguese. Who knows what happens after that?
Quite the opposite. So German was 6:30 to 8 in the morning, 5 days a week for years. And and Portuguese, I was already working after my undergrad, so it was on my lunch break. And those things ended up helping me because I did part of my PhD at the German Aerospace Center, Deutsches Luft- und Raumfahrtzentrum, just to show off the German. Yes, it did. And that was such a cool experience because then my wife and I got to go live in Germany, and speaking German was very helpful, right? Although it could have been more. Once you're there, like, and you start speaking in German, you're like, oh, your accent, you speak English, let me practice my English, and you don't get to practice your German. But you're being there, it helps a lot, right? And same thing with Portuguese. The very first thing I did after graduating of my PhD was I went down to Universidade Federal de Minas Gerais in Brazil, funded by the Brazilian Space Agency, to train students and teachers and professors on how to do space projects for students. And this came from my extracurricular work at Universidad del Valle de Guatemala, which is my alma mater, developing, just helping students, undergraduate students, develop the first satellite of the country. And that launched last year and is currently operating.
So you did this work in all over the world. This is so cool that you're everywhere. What has this? I imagine it has, but working in all of these different agencies and all these different countries, what impact has that had on your career and also personally? Is that something that like your education kind of just fell into place as life happened? Or when you said you learned German and Portuguese, then did you say, well, now I have to go to Germany and Brazil and use these skills that I have, and that led to you doing what you did there?
All right. Well, I think that once you learn a language, you're just so much more curious about the culture, and then
Sure.
you want to go there. So it didn't feel like I had to. And the other thing is, I don't know, but I think I'm very passionate about international collaboration. And since what I do is science and engineering, that's what I like trying to implement it on. But the more we know each other, the less likely it's going to be that we're going to get in trouble with one another. And if that's why I really like International Space Station, for example, is like all these countries, even when there are political problems on Earth, space station is still orbiting, cosmonauts and astronauts and people from all over are like working together as humanity, because that's by the end of the day what we are, right? One species.
Yeah, I recently saw a quote from Leland Melvin, who was talking about being in space. And he said, comparing it to here on Earth, we have to work together or we die. If you're on the space station, if you don't work together, you die. And it's
Mm-hmm.
the same thing here on Earth.
Yeah.
So you said that you have come to the US now, you're a citizen now. So congratulations on that.
Thank you.
Do you plan on staying here for a while, or is this kind of global research more your thing? Would you like to travel anywhere else or go study other places?
Oh no, totally settled roots here. My wife and I have a family here, and I'm very happy with all the opportunities I have at BioServe and the University of Colorado. Not only can I support other scientists' experiments going to space, but now I have my own, like the one like space biofilms and like the one going around the moon. It's called Deep Space Radiation Genomics. And yeah, this is the place to be for so many reasons. And really, I think that's true for a lot of immigrants. And it's also important if try to never forget where we come from. And that's one of the reasons too for which I like international collaborations, trying to give opportunities to other people, which is really mutually beneficial. For example, if you see on my webpage that my projects, they all have an international aspect to them, and it is mutually beneficial in that they have the opportunity to work on space-based science, and from the other hand, they bring knowledge that either me personally or my team doesn't have, and so it's complementary. So we have more powerful science by working together through these collaborations.
That makes good sense, and it's kind of not, I don't want to say hopeful, that sounds like too drab, but it's inspiring to see people such as yourself passionate about making sure that this collaboration continues to happen and pursue it as one of the primary goals of your work. So thanks for doing that.
No, thank you. That's a very nice thing to say. I appreciate it.
So the question that we like to wrap up with a lot is, and you've actually touched on this several times, which is awesome, but I'll ask you to kind of summarize everything that you've said in this vein. What advice do you have to young people who, after listening to this, say, wow, that Dr. Zia guy is awesome and hilarious and sounds like he's having a great time and I want to go do that? And so
Yeah.
they say, okay, I know I'm into bioastronautics. What advice, if you could wrap all of those different tidbits of advice that you listed over this last hour, what advice do you give to young people who now want to pursue this as a career?
You say you want me to wrap it or wrap it? You want me to like freestyle right now?
If you can wrap it, that would be awesome.
That would be too hard, Colin. So I'm just gonna go with non-wrap. I'm gonna wrap it.
Okay, okay.
So it depends on the age. If it's elementary or middle school or even high school, I would say grades matter. See, I always thought, why is my mom giving me such a hard time about my grades? Who cares about the grades? You will in your future. It opens doors. You know, these movies where you have this magical key that opens this magical door to who knows where? Those are your grades. Extracurricular activities. Once more than one person is applying to a position, to either a job or grad school, you're going to be compared against in a quantitatively way, meaning that you want to make yourself stronger a case than others. And one way you can do that is through extracurriculars. And it can be based on what it is that you want to do. It can be languages. If you want to go into computer stuff, then you know different software languages or different software. No, so yeah, this kind of extracurricular, start a student group, these kind of things. Volunteering, I think it's very important. But really, you got to do it if it comes from the heart. But there's so many different things in which you can volunteer. We are always more fortunate than a lot of other people. So finding time to help others, it's pretty important. And then if you're older and now you're kind of like at college, you're getting so old. Then same thing of grades and extracurricular. But don't forget the multidisciplinarity of real life. And it's important to know more than one trade. It's going to help you a lot. Stay positive and keep working hard.
I think that is a perfect way to wrap up the episode. Dr. Luis Zia, thank you so much for joining us today. That was absolutely a pleasure to talk with you.
Likewise, thank you for having me. It was really nice talking to you, Tara and Colin.